Hello people, we're going to be talking about Staphylococci in this video, and this is the part 1. It's going to cover Staph Aureus only, and this picture you see tells you that they are grape-like clusters under the microscope. So let's get started. And there are 3 main species that are medically relevant: Staphylococcus aureus, Staphylococcus epidermidis, and Staphylococcus saprophyticus. Aureus is the main one, and I'm going to be talking about Aureus in part 1, and the rest 2 are going to be in part 2, so stay tuned for that. Alright, so a few properties here. I give the credit to Kaplan Medical. There are 3 species, as you can see, and there are some various differences. For example, Aureus is coagulase positive, and it's hemolytic, beta hemolytic to be specific. And the other ones are non-hemolytic. So please pause the video, jot down these points, remember them, do not memorize them, try to understand them, and then, hopefully you'll understand things better. Alright, let's get started with Aureus. Right, most commonly causes bacterial conjunctivitis, commonly associated with hospital-acquired pneumonia, septicemia, and surgical wound infections. It's also an important cause of skin infections like cellulitis, folliculitis, and impetigo. It produces staphyloxanthin. It's a pigment and gives its colonies characteristic golden color. It also neutralizes reactive oxide species in the neutrophils. It's virulent. Also produces hemolysins, and they hemolyze RBCs, producing iron, which is a source of energy for this organism. And 90% of the strains produce beta-lactamase, okay? And they're plasmid encoded MECA gene for PVP alteration. That's located on chromosome. And then these strains are commonly called MRSA and NRSA. MRSA stands for methicillin-resistant Staph Aureus. NRSA is nafcillin-resistant Staph Aureus. MRSA is 50% of Aureus strains, so that's a lot. And it's usually found in hospitals and hospital patients, hospital workers. The most common one is USA 300 strain. And cassetta genes, which is found in Enterococci as well, encodes vancomycin resistance, okay? And these are located in a transposon on a plasmid and encode the enzymes that substitute D-lactate for D-alanine in the peptidoglycan. Alright, very important cell wall components and antigens. Protein A is a major protein in cell wall, and it's a virulence factor because it binds to Fc portion of IgG at complement binding site, and so therefore it prevents the activation of complement, ultimately leading to reduced opsonization of bacteria. Teichoic acid is a polymer of ribitol phosphate and mediates the adherence of this bug to the mucosa. Polysaccharide capsules, there are 11 serotypes. Type 5 and 8 are the causative agent for more than 85% of infections. Some strains are coated with small amount of polysaccharide capsule called microcapsule, and capsule is poorly immunogenic, so it's not useful in labs. Surface receptors for specific bacteriophages permit phage typing, okay, in epidemiology. A peptidoglycan has endotoxin-like properties like activation. It's responsible for activation of complement and coagulation cascades and cytokine production by macrophages. Nose is the main colonization site. Skin in hospital personnel and patients, vagina in about 5% of women also harbor this organism. Hand contact is the most important mode of transmission. Fomites and sheddings from human lesions are important sources. Now, predisposal. These, diseases by Staphylococcus aureus are favored by a heavily contaminated environment and compromised immune system, particularly the humoral immunity. Plus, diabetes and IV drug users and chronic granulomatous disease patients are also prone to the infection by Staph Aureus. And try to remember the last video that I posted. It's pathogenesis. If you remember the stuff from last lesson, which was on pathogenesis, you'll be fine. Okay, so toxin production and induction of pyogenic inflammation. That's pretty much it. Oh, and by the way, before moving ahead, the cause of disease was by toxin production and induction of pyogenic inflammation, and that was a pathogenesis. Anyhow, toxins and enzymes. Toxins and enzymes. Well, a few important toxins you see here: enterotoxin, TSST, that's toxic shock syndrome toxin, exfoliatin, and several leukocidins, for example, alpha toxin and PV leukocidin, that's pentan valentine leukocidin. Few enzymes are coagulase, fibrinolysin, protease, hyaluronidase, nuclease, and lipase. We're going to go into detail right about now. Alright, what I'm about to teach you in this slide is important for the next slide, so be attentive. Wake up. Enterotoxin causes food poisoning. Food poisoning characterized by prominent vomiting and watery, non-bloody diarrhea. It acts as a superantigen within the GIT to stimulate release of large amounts of IL-1 and IL-2 from macrophages and Th cells, respectively. Cytokines released from lymphoid cells stimulate the ENS, that's enteric nervous system, to activate vomiting center in the brain that's going to lead to vomiting. And then enterotoxin is heat resistant. Also, it's resistant to stomach acid and jejunal stomach enzymes. 6 immunological types of enterotoxin, types A to F. Okay, so just TSST causes toxic shock syndrome, especially in tampon using menstruating women or individuals with wound infections. Also occurs in patients with nasal packings to stop bleeding from nose, and is produced locally by Aureus in vagina, nose, or other infected sites. Now, toxin enters blood, leading to toxemia, and blood cultures typically don't grow Staph Aureus, so that's important. Remember, because that's going to be helpful in lab diagnosis. TSST is a superantigen, right? So releases IL-1, IL-2, and TNF, and they are the culprits. That's the easy way to remember it. About 5 to 25% of isolates carry gene for TSST. Okay. Exfoliatin causes scalded skin syndrome in young children. It's epidermolytic and acts as a protease that cleaves the desmoglein in desmosomes, leading to separation of the epidermis at granular cell layer. And several leukocidins, alpha toxin, which causes marked skin necrosis and hemolysis. PV leukocidin, interesting one, is a pore-forming toxin, killing cells, especially WBCs. Okay, there are 2 subunits of this toxin. They assemble in cell membrane to form pore, through which contents leak out. Genes encoding PV leukocidin is on lysogenic phage. PV leukocidin's virulence is indicated by severe skin and soft tissue infections. Important: severe soft and skin tissue infections caused by MRSA that produces PV leukocidin. Severe necrotizing pneumonia is also caused by PV leukocidin producing strains. So about 2% of the clinical isolates produce PV leukocidin. Not as important, but clinicals of this PV leukocidin thing are interesting. Enzymes, we've already mentioned them, but few, for example, coagulase, clots, right? And thus walls off the infectious site wherever there is infection, and that leads to retarding of the neutrophil migration into the site. Staphylococcus, for example, is a fibrinolysin. Okay, staphylococcus, remember that, is a fibrinolysin that lyses the thrombi. Clinical diseases by Staphylococcus aureus are categorized into 2 categories: pyogenic diseases and toxin-mediated diseases. Remember that generally which symptoms we see in patients is dependent on a specific strain of a specific bacteria and its properties and virulence factors. In pyogenic diseases, skin infections include furuncles, boils, carbuncles, and impetigo, paronychia, hidradenitis suppurativa, and mastitis. Remember that lymphangitis on forearm is associated with infection on the hand, and there are some severe skin and soft tissue infections by PVL-producing strains, and they're typically community-acquired, therefore very common in homeless and IV drug abusers. Septicemia can originate from any localized lesion, especially a wound infection or IV drug abuse, and clinicals and the symptoms are similar to those of gram-negative bacteria, for example, Neisseria meningitidis. Endocarditis on normal or prostatic valve, especially right-sided tricuspid valve, is common in IV drug users. Osteomyelitis and arthritis may be introduced locally at wound site or by hematogenous spread from distant sites, and it's very common especially in children. Post-surgical wound infections are also common. Pneumonia in post-operatives is also common. Staph pneumonia often leads to empyema or lung abscesses. In many hospitals, it's the most common cause of nosocomial pneumonia. Nosocomial pneumonia in general, and especially ventilator-associated pneumonia, is usually found in ICU, intensive care unit. Community-acquired MRSA causes severe necrotizing pneumonia. Okay. Conjunctivitis typically presents with unilateral burning eye pain, hyperemia of conjunctiva, and purulent discharge. Is transmitted to the eye by contaminated fingers, and Aureus is the most common cause overall. Abscesses, finally, in any organ actually, could spread from bacteremia, and therefore they're called metastatic abscesses. 3 toxin diseases: gastroenteritis caused by enterotoxin, which is preformed in foods, and causes vomiting and diarrhea. Vomiting is usually more prominent. TSS, toxic shock syndrome, characterized by fever, hypertension, diffuse macular sunburn-like rash that goes on to desquamation, and involvement of 3 or more organs like liver, kidney, GIT, CNS, muscle, or blood. Scalded skin syndrome is characterized by fever, large bullae, and erythematous macular rash, large areas of skin slough, serous fluid exudation, and electrolyte imbalance, plus the nails and hair loss is also possible here. Recovery is usually within 7 to 10 days, and this syndrome most often occurs in children. Alright, Kawasaki syndrome has unknown etiology, and we're talking KS syndrome over here because several of its features resemble TSS caused by superantigen of Aureus. So it's a disease of children, young children, younger than 5 years old, and it's more common in Japan. Therefore, it's more common in children of Asian ancestry. It's characterized primarily by fever of at least 5 days duration, bilateral non-purulent conjunctivitis, lesions and edema of lips and oral mucosa. Characteristic strawberry tongue is observed in this exam, and erythema and edema of hands and feet that often ends with desquamation and cervical lymphadenopathy. So these are characteristics. Just try to remember these for clinicals. Now the main cardiac involvement, like arrhythmias, myocarditis, and regurgitation involving mitral and aortic valves, and aneurysm of coronary artery, are most significant clinicals that might prove fatal. So, okay. And finally, lab diagnosis. Smears are taken from lesions, and those lesion smears reveal grape-like clusters under the microscope. On the agar, however, the colonies are golden yellow and they're beta hemolytic. And the usual choice of agar is mannitol salt agar, and there's no serology for acute infections. For TSS, as long as clinical criteria is met, Aureus isolation isn't required. And lab findings of TSS are usually isolation of TSST strain and antibody to toxin during convalescence, during the convalescent period, though it's not useful for diagnosis in acute infection. So just clinicals are enough, eh? Okay, so how do we treat this bad boy? Remember that 90% or more Aureus strains are penicillin G resistant. So that's due to beta-lactamase production. So in that case, we treat this with nafcillin, methicillin, or cloxacillin, and then or some cephalosporins. But there are some strains that are resistant to these drugs too, like MRSA and RSA. And then we use vancomycin, and then there are some vancomycin-resistant strains, and we use daptomycin there. Clindamycin and or trimethoprim sulfamethoxazole, I think, are used in non-life-threatening MRSA and NRSA. For TSS, we give plenty of fluids, inotropic drugs, suppressor drugs, and removal of tampon and debridement from infected site. Mupirocin is very preventive. It's usually used in prevention and prophylactic type of drug, and it's usually topically used for skin lesions, and incision and drainage is required for abscesses. And the last slide is about prevention. There's no vaccine as of now. Hygiene is extremely important. Hand washing and, keep yourself clean. Removal of shadows from high-risk areas. And cefazolin is used preoperatively to prevent surgical wound infections. And good luck for the exam, and thank you for watching. Bye-bye.